I will start sharing my screen. Can you see it now? Laura? Okay, great. Then I will start with my talk. Laura, many thanks for introduction. I will focus on measuring plant physiology. And here one of specific points what I would like to discuss, new result what we discovered during the project runtime, what we called biofeedback-based agriculture. So it's a new feature what was developed in the project, and how do we see this in application also in different areas. So, and of course I will also talk about air pollution measurement what we have done. I would like to start with a short motivation why this is really important and why we are doing this. And here, one of main ideas is that plant have a huge adaptation potential. This adaptation potential was developed probably during the evolution to enable surviving in different conditions. And one of, let's say, interesting consequence of this high adaptation potential, if all requirements that plant needs are met, plants can achieve a huge productivity. Just to give you one example, in the wheat production, the natural productivity of wheat in the field condition is about 3 tons per hectare. But if all these requirements are satisfied, plant can reach about 2,000 tons per hectare. So this is theoretical maxima. And just think about it, for instance, we can reach 600 more food from the same area. This has a huge commercial potential, a lot of absolutely the new challenges what we see. And all this development currently, they go in 2 directions. So one is really increasing productivity, and in, for instance, modern results in 10-layer vertical farms, currently we can reach something like 700 tons. I mean, 200 times more than in natural conditions. This is really great. But problem is that we produce more food than we consume. So from this point of view, food distribution is inhomogeneous, and this is a problem. And therefore, it generates a second developmental line, this food production in areas which are impossible for food production. As for instance, Arctic, Antarctic regions, deserts, and currently new developments are space application. A lot now in progress for instance, for developing such a model for moon station, potentially in Mars application. So there is, so this area, they have a great scientific potential. So to give a short overview about, let's say, commercial impact or commercial dimension, what are behind this. We have been about 1 month ago in one of the largest exhibitions in Hanover related to agriculture production, in particular to in-house farming. These are very different types of farms, microbial farms, farms with vegetables, with insects. And of course, I mean, large number of companies, of ideas, of innovations. Just to give some numbers, I mean, to characterize this area, we see, for instance, 3 main driving forces, with a total market something like 100 billion per year. I mean, this is one of the largest market ever. And of course, I mean, there's a lot of commercial potential, of money, of investor. I mean, this is quite inspiring. I mean, participation in this event is quite inspiring, and just I mean, demonstrate a lot of commercial, of companies, of industry behind all this development. Okay, back to the idea how to achieve high productivity and high adaptability. So key point here is that plant have to define our environment. I mean, what does it mean? For instance, if plant need more light, plant should switch on light, not a farmer, not a periodical timer. I mean, plant defined if there is a need of light, okay, switch on light. If, for instance, a plant need more water, it should switch on pump and receive more water. So this is more or less main idea. We call it biofeedback-based approach. So we are measuring by signals from plant, from physiological stage, from soil, from environmental parameter, process with different methods. For instance, artificial intelligence is also applied here, and then use it to control different phytoactuators. Currently, we think here on this site is about 15 to 20 different devices, including light, water preparation, air preparation. I mean, there is different approaches here as well. So, and the whole system create a biofeedback, and this biofeedback-based system can be used as not only in agriculture but also in, for instance, estimating water quality, measuring water quality, what I will show later. Here, one of, let's say, important point, the system is rather complex. So plant is extremely complex biochemical machinery, as a complex organism, and if we apply this technological cycle, we need models. So we cannot work any longer with empirical data. We need some model-based approach here. And to use this model, we involve fluidic movement in plants. Just to understand what does it mean, it's very comparable with a blood system of humans. So if we are measuring, for instance, blood pressure, if we are measuring sugar in blood, if we are measuring some biomarkers in blood, we receive information about physiological stage of human. In the same way, if we are measuring hydrodynamic parameters of plant, so water movement and similar issues, we receive information about stage of the plant. In more detail, we have here, for instance, very this low area. It has information about water, about irrigation, about chemical content of water. In the upper area here, we have information about, for instance, transpiration, VPD, environmental pollutants in air, light, fetal light, and similar parameters. And here we have in fluidic system also a lot of organic components from photosynthesis. Here in the middle area is a lot of physical effects related, for instance, to capillary forces, to xylem employing tissues. So in this way, the whole model is quite complex. It still involves different parameters from environment, from plants. And now if we characterize these parameters, we can, usefully work with this, for instance, for controlling, for making some conclusion, estimating stress and different issues. So now in turn, here we have a huge challenge. How to measure all these different physiological parameters, because here is a key issue to be able to measure all different parameters in different regions of the stem. State-of-the-art approaches, typically we have some classical, let's say, methods like dendrometer, Tugor, that's measuring more or less some mechanistic parameters, for instance, how thick is the stem or changes in the stem diameters. Well-known are thermal methods for heat balance, heat impulse is a classical approach for subflow measurements, as the electric methods as well. We focus on two new methods that was proposed last years. One is related to electrochemical impedance spectroscopy, and the second one a nuclear magnetic resonance. And the whole talk later on is focused on these two methods, once again to achieve a high adaptivity and high productivity of plants. So, now I briefly explain what are these both methods and what kind of result can we expect, and then go to results what we received with these measurements. So electrochemistry generally is measuring current response, so to electrode they create potential, and we measuring current output here. Since ions are they carry charge, In this way, by measuring current output, we are measuring ionic content of tissues. Like I said, here in the lower area, there is more ionic-rich fluids from minerals, from soils. Here in the upper area is more organic substances with less ionic content. So we easily can differentiate, for instance, upflow and downflow from xylem and phloem tissues. We can see the aerodynamics. And here there are two very different approaches how to do this. So first we can measure over frequencies. So here is the idea that ionic response dependent on chemical, let's say, elements that are behind of the ionic flow, ionic content, and so we are able to, for instance, to see what sort of ionic content is in the fluid. We can see, for instance, changes in the ionic content. We can see differences between organic and high ionic substances. So we can perform more or less analysis on ionic level and ionic groups. The second approach, if we are measuring over time, so in this case, for instance, we fix our frequency and some ionic group what we are interested in, and do this timely, so we record a dynamic of these parameters. In this case, we receive so-called circadian rhythm. All plants, they perform in very highly periodic way. And here we see, for instance, dependency between light and subflow, between irrigation and transpiration. So this, all these parameters are absolutely well visible in this dynamics. We see, for instance, water deficit strategy. If we apply, we see how we change water content in stem. I mean, this measurements mode carry a very extremely useful information. So this is about electrochemical approach. Now about nuclear magnetic resonance. Generally, this technology is one of the most useful approach in the chemistry. It can identify different substances based on these resonances, spin resonances, and chemical shift. So, we know application of this nuclear magnetic resonance effect, MRI technology in, for instance, tomographs for medical purposes. With plants, the whole development started about, I would say, 10 years ago. And this is the same idea. So we can use this for identification of different molecular substances, use in metabolomics, used to identify different processes even on the cellular level. There are two different developments for MRI technology. The first one using permanent magnets. Now it's more or less standard in some portable MRI scanners. And the second one is used magnetic field, earth magnetic field. And the difference is, with permanent magnet field, extremely heavy devices, for instance, between 5-6 kilogram up to 40-30 kilograms. So it's not really like a portable sensor. You need a holder, you need some fixing elements for keeping the sensor on plants. Though from this point of view, this earth magnetic field sensor are much more useful. Another point, here is a working frequency. The resonance frequency and hydrogen, so in this kind of sensors, about 50 to 60 megahertz. Here in this case, we have only 1.5 kilohertz. So here it's much more useful. So we focus also primarily in this type of development. Another issue is usability of sensors. For instance, here the sensor is only a coil on a plant, so it is extremely, reliable. Like electrochemical sensor, we have only 2 needles inserted. So from this point of view, it's so both sensors are extremely reliable. Can be used in field conditions. And maintenance effort for maintenance is extremely low. And by the way, both sensors are quite cheap. So now, what kind of information can we receive with MRI technology? First one is resonance. So it's classical for MRI. So we receive resonance for hydrogen. In the same way, we can receive resonance for carbon. And here we also see chemical shifts, so we can identify molecules that have, for instance, hydrogen or have a carbon. So it's a large number of different molecules that can be identified. And we, as a non-invasive technology, we can see this in different position in plant stem, in plant tissues, even, like I said, on the cellular level. In application with plant, this technology is, let's say, has a specific direction. Like I said, almost everything in plant science is related to water, to hydrodynamic system, and MRI here also focus on water. For instance, here we see water content in different tissues. So, for instance, this only psyllium and chloem is only small tissues around the stem, and we see most of water is in this tissue. Exactly, we can see a special distribution of water content. We can see, for instance, in each of these points, dynamic of water. So, if we focus on dynamical aspects, we see the same way this hydrodynamic circadian rhythms in plant. So here, one important point that MRI, so if we focus on MRI technology and water content, we receive identical result with electrochemistry. For instance, here circadian rhythm, we see the same circadian rhythm here with electrochemical measurements. And this is important because MRI technology is used currently in plant science on, I would say, something like 5% of its full potential. And the reason is MRI is not not measuring a water content by itself. It's measuring only one of isomers. So water has two spin isomers, ortho and para isomers, and para isomers is not sensitive to magnetic field, only ortho isomers is sensitive to magnetic field, and MRI is measuring only isomers, one of isomers. But there's a second variable is the water content. So it could be, for instance, more water inside of tissue or more isomer inside of tissues. So there are two variables, and to use this in water science, people usually say, well, we assume there is a specific dependency between ortho and para isomers, and so we can approximate water content. So just to understand correctly, so all this data is only approximation of water content. And in this case, people skip all this quantum phenomena, spin phenomena that MRI is developed for. So in this way, in plant science, so not a full potential of this sensor is used. To avoid this, our approach is basically to involve electrochemical sensor, which will be defined water content. So we define water content by electrochemistry, and then by MRI we can really focus on spin phenomena. We can work on the level of quantum chemistry, spin catalyst, and all this fancy stuff. What is basically this MRI technology is used for. So this main idea. Now a little bit about our results. This is. This sensor is used already a long time. This is a new generation with two needles here. We also can have different working distance, but we say well, it's about 10 millimeters. So we are measuring water contents in the area of 10 millimeters of stem. And here is the MRI. This, like I said, is focused on Earth magnetic field without permanent magnet. So here inside only a coil. We use some engineering approach to put sensor sidewise, not axial-wise, but sidewise. We lose a little bit of homogeneity of magnetic field, but still, I mean, for plant working volume about 2-3 millimeters, so it's absolutely enough. And here again is a prototype because as a new, we still have some sensitivity issues because Earth magnetic field is extremely weak. Now, how can we use all these results in plant size? We return back to hydrodynamic model, and I simply say why currently this plant grow is not efficient. We can easily see with these approaches. Here is a water content in tissue. And we see in the nighttime water is accumulated in tissue. Here this light is on, this daily period, night period, daily period. So light on, light off. And here in the morning time, light is on, transpiration start, photosynthetic activity start, and plant actively consume water from stem. And here we discover some point it's species dependent. It's we cannot generalize to all species. Each particular species behave differently. But we see in many cases this point B, it means for transpiration not enough water inside of stem, and plants stop transpiration. We see this exactly the same data in the transpiration sensor, and plant decrease photosynthetic activity. So it means to some extent plants switch off and waiting until water is again accumulated in stem. And here this time point is D, somewhere in the night time, and here plant is ready to start photosynthetic activities, start to work, but it's a night, there is no light, and plant is waiting to the next morning. So it means from this 24-hour cycle, plant is used something like 30% of active time for production activities. So it means the current grow technology are extremely inefficient, and this explains this low efficiency in natural conditions more or less, and high if we use, for instance, adaptive technology, we can dramatically over 200 times improve productivities. So the idea is rather simple. So for instance, here in the point B we switch off light, here in the point D we switch light again, and so plant instead of 24 circadian rhythm, they will have something like 6 on light and 4 off light, then something like 6 or 7 on light, and so they develop our circadian rhythm and they control our environment. And in many cases, we even do not see a periodic circadian rhythm. So it depends really on developmental stage of the plant. So it's extremely impact productivity. So here are some visual examples, P and wheat. The biomass grow if plant behave adaptively is really dramatic. In 2 days, instead something like this, you see a huge biomass increase. In the same way, we can work with irrigation. So we see water content in tissue. So we can see how far it's propagated through the stem. And here is example from hydroponic. The hydroponic, so this type of hydroponic has a periodical irrigation every 2 hours, and typically farmers use a periodical timer, for instance 1 minute irrigation every 2 hours, but we see that this approach is extremely inefficient again, because in some cases plants are over irrigated, so we have high losses of water. In other cases, it depends on developmental stage, irrigation is definitely not enough, and we have increase instead of, for instance, 1 minute, we have to irrigate 5 minutes, something like this. And so if we do this feedback-based approach, we can see position of, for instance, enrichment of water in plant tissue, and based on this, perform adaptive irrigation. This again increase essentially biomass production. How it look like in our, let's say, test facilities, we developed this vertical farm. It's not a classical vertical farm, just to say from the beginning. This is, at least I'm from robotic field, and I see here all this what we know about robotics. This is a complex robot. Here we have in this system, we have sensors, also multiple sensors, we have different actuators, we have information processing unit, we have user interface to users. So it's a highly complex system, so it's a robot more or less. And here, for instance, some results in microgreens. For instance, microgreens is a 1 day under light. You see this not even green, it's some yellow of color, and we receive microgreens in 2 days. We receive ready to use microgreens in 2 days. This is P very young, is a 1 day, and this next is a P about 5 days. So you see a huge development. And there is wheat. I think this with about 1 week, and this about 2 or 3 days. So this extremely fast development. I receive almost always many questions how to do this practically. I mean, there's tiny small stems, how to put electrodes. Like I said, all this technology, they're extremely simple, as or at least primary sensitive element are simple, and in this way reliable. Here we use, for instance, we call it measurements plant and productive plants. For measurements plant, we just insert needles in the stem in several positions, sometimes only one position, sometimes 3-4 positions, depending on species, and perform measurements, long-term measurements. In production, we change every 3-4 days these production spaces, but these measurement spaces, they can really be a long time in the vertical farm, and we basically use this plant for measuring parameters of water plant. So it's a terminology of homogeneity. So in this case, we have a high homogeneity of a system. Some numbers again, like I said, microgreens, I mean, currently what we see is extremely fast, only 2 days. Generally, all these vertical systems, they work in cycles, and if we started with this as about 1 year ago, we used typical 6-7 days cycle. Currently, we work on 4 days cycle for all species. Biomass we are measuring now, but at least we see in some cases we receive definitely more than 30% of biomass increase. Now think about, for instance, commercial application of this technology. Energy, but energy currently not a point. There was our initial assumption that energy is a point, but biomass. So we can, we keep the same energy consumption, but we increase biomass production. Another interesting point is water, because what we learn, water is extremely important in countries like Arabic countries, desert, North Africa, even in Spain. What I learned in summer region, there is problems with water, and so farmers use water deficit irrigation, so just reducing our water. And give water, also keep on irrigation only in specific time moment, just to save water, because there is not enough water. And here, one of issues what we saw, if, for instance, here as example, we apply here deficit irrigation, and we see decreasing of circadian rhythm, and basically this plant, they die in 2 weeks. So, but the process that lead to dying of plant, we see even now. So after 1 week, we see that circadian rhythm. You see here normal circadian rhythm, and now plant is losing circadian rhythm. It means the hydrodynamic system is highly disturbed, and plants start to suffer. And so it's rather easy. So by using this biofeedback, we can monitor water distribution and all these periodical activities based on water. And if we see, for instance, starting of degradation of hydrodynamics, we can put more water, or we can switch a strategy. We can work purposefully with this water irrigation. So it is one of the really nice approaches that probably has potential. Another issue, like I said, and this was one of the main ideas in WATCHPLANT project, that plant physiology is extremely sensitive to environments, and we can use it for pathogen detection. So not only pathogen, but generally stress detection. Stress can be from water, even from sun, that also create stress. So we are able to detect it. And here is the same way. If we are measuring normal periodical activities of plants, so we know dynamics. So we learn, as first we need to learn dynamics of plant in the normal condition, and then if we apply different stresses, we are able to differentiate it and to characterize it. We work mostly with ozone, but in the same way with microdusts and with pathogens, also fungal pathogens and microbial pathogens. In all cases, we see extremely evident reaction. So here is example with ozone. We use indoor setup and outdoor setup. They have different analysis, but still, we are able to detect ozone at extremely high, as an extremely low level. For instance, here is only 15 ppb. So per billion. We see here that hydrodynamic system of plants start to react. So we see closing of stomata and changing of hydrodynamic behavior of a plant. So in this case, we are able to say, okay, well, the stress start And if we use multiple plants, for instance, here, like in this case, we have quite good relation between false positive, so we have about 7% of false positive, and true positive is about 92%. So it is quite good result in this case. Like I said, this approach can be used for pathogen detection. We think now about fungal pathogen detection because it's quite important in wheat, for instance, production is quite important. So we think they have a specific, also not only, let's say, a reaction, but they have specific dynamics. So, and we can detect this specific dynamics. So the hope is that we are able to identify this pathogen. So this is last slide. I think this is for future. We need to mention that this quantum biological aspect is become more and more relevant, quantum biology. And here I repeat again that by combination of electrochemistry and MRI, we have in fact a combination of chemical side and quantum side. And here is important because, like I said, water has two isomers. But a relation between two isomers that define physical and chemical properties of fluids. So they change, also first of all, they change chemical reactivities. Same water can be more reactive or less reactive. So we extremely see this in plant. It change viscosity. So viscosity is capillary forces, so the different speed of movement in stem. It's only by a combination of these two isomers. So we see changes of heat capacity, and this explains why water treatment strategies, for instance, hydrodynamic cavitation, enrichment with microbubbles, oxygen microbubbles, but with hydrodynamic cavitation, magnetic processing of fluids, they have a huge impact on plants because exactly in this case we change combination between para and ortho isomers. So in this case, with combination of both sensors, first of all, we are able to control the physical processes in plant stem, and we have access to quantum side. And here is important, once again, the sensors are, they are not super cheap, but they are not expensive. So because they are quite simple, basic elements is quite simple. So in this case, we receive a kind of extremely affordable technology for exploring quantum side of quantum biology. Let's present this way. So I would like to conclude. What we saw that first of all, this hydrodynamic modeling, we started this with WATCHPLANT 2-3 years ago. It seems is quite useful, and we publish it, and we receive currently feedback, so people really like it. So it's extremely useful. We think if we use a biofeedback-based and hydrodynamic, it's quite promising. A new approach, definitely new approach in precise agriculture. And here some emergent, once again, effect here, we involve fluidic analysis that was not in the plant biology. So it's completely different from physical chemistry. Now it's in plant size. We see new sensors. From our point of view, combination is useful, as electrochemical and MRI. If we, for instance, focus on the hydrogen resonances, but in the way in carbon resonances, so both are possible. And in this case, we use measuring water content, and we usually proton density of ortho isomers. So we work with quantum biological aspects. And you know, quantum biology now is a huge field. For instance, spin catalyst, spin chemistry, and with these approaches is a future project probably. So we are able to work in this side with plants. From practical point of view, what we saw as a biomass production is extremely useful. A lot of results here. And another point is extremely also interesting is working with water deficit, because many countries suffer of, let's say, with climate change, less and less water. So we are able to work with this water deficit irrigation in a very controllable way. So this end of my talk. I would like to acknowledge, first of all, thank you for European Commission for funding. This is extremely important, especially now. I would like to thank all members of our team and WATCHPLANT Consortium. We had a lot of technical discussion, phytosensing discussion, so many thanks. So thank you.